Hello dear students! In this video, we're going to speak about microscopes. Let's start. What is microscope? A microscope is used to produce a magnified image of an object or specimen. Anton van Leeuwenhoek was the first to invent a microscope powerful enough to explore the world of microbes. His discovery stimulated an explosion of interest in the scientific use of microscopes. Since the 18th century, many new types have been invented, of which the most commonly used today are the compound light microscope and the electron microscope. The compound light microscope. The compound light microscope is also called a light microscope or optical microscope. The lens refracts the light to give a magnified image of the object. The image may be projected directly into the viewer's eyes or into photographic film. A photograph taken through a light microscope is called a photomicrograph or a light micrograph. Magnification and resolution. The magnification of an instrument is the increase in the apparent size of the object. The total magnification of a compound microscope is worked out by multiplying the magnification of the objective lens by that of the ocular lens. There is virtually no limit to the magnification produced by a light microscope. It depends on the power of the lens used. However, above a certain magnification the image becomes blurred and it is impossible to distinguish structures lying close together. This limit of effective magnification is called the resolving power or resolution of the microscope. It is defined as the ability of a microscope to show two objects as separate. The resolving power of the light microscope is limited by the wavelengths of light. Light microscopes can magnify objects up to about 1,500 times without losing clarity. The electron microscope. Electron microscopes use a beam of electrons instead of a beam of light. Electron beams have a much smaller wavelength than light rays, so electron microscopes have greater resolving powers and can produce much higher effective magnifications than light microscopes. There are two main types of electron microscopes: the transmission electron microscope and the scanning electron microscope. The transmission electron microscope. The transmission electron microscope is used to study the details of the internal structure of cells. Extremely thin samples of the specimen are needed. To make this, the specimen is supported in a resin block to prevent it collapsing during cutting, and is sliced with a diamond or a glass knife. The section is then impregnated with a heavy metal stain, such as osmium tetroxide. As the beam passes through the specimen, electrons are absorbed by heavily stained parts but pass readily through the lightly stained parts. Electromagnets bend the electron beam to focus an image onto a fluorescent screen or a photographic film. Photograph taken through an electron microscope is called an electron micrograph. The most modern TEMs distinguish objects as small as 0.2 nanometer. This means that they can produce clear images magnified up to 250,000 times. The magnification is varied by changing the strength of the electromagnets. The scanning electron microscope. The scanning electron microscope is used to produce three-dimensional images of the surface of specimens. Electrons are reflected from the surface of a specimen stained with a heavy metal. This enables the scanning electron microscope to produce images of whole specimens, cells, tissues, or even organisms. Although electron microscopes have revolutionized cell biology, they have not completely replaced light microscopes. Light microscopes are used to examine living and unstained specimens. Preparation of specimen for electron microscopy is complicated and time-consuming. Electron microscopes are very expensive and can be used only to study dead specimen stained with heavy metal, which might well produce artifacts. That is the end of our video session. Thank you for watching.